Hello, everyone. My name is Tim Zhou, and I am a postdoctoral fellow at the University of Wisconsin-Madison, working with the Ravel Lab in collaboration with the Camp Lab. It is my pleasure today to give today's presentation titled Transendocardial Injected Human Pluripotent Stem Cell-Derived Cardiac Progenitor Cells Engraft and Differentiate Without Associated Tachyarrhythmias in a Swine Ischemic Cardiomyopathy Model. Dr. Camp is a consultant for Fujifilm CDI. Doctors Ravel and Schmuck are co-founders of Cellular Logistics. Fujifilm CDI and Cellular Logistics provided partial support for the study. Coronary artery disease imposes a significant burden on our society, contributing to an estimated 1 of every 7 deaths in the United States. The global prevalence of coronary artery disease has increased by 21.1% over the past decade, with an annual associated cost of around $213.8 billion. Coronary artery disease is problematic in part because it can result not only in heart attacks, but also heart failure. 16% of men and 22% of women who suffer a first-time heart attack develop heart failure. In Europe and the United States, coronary artery disease is the most common cause of heart failure. This has resulted in a rise in the number of heart failure cases. Current therapies for heart failure include pharmacologic neurohormonal blockade to limit adverse remodeling. Despite these options, heart failure is a progressive and terminal diagnosis, which will necessitate transplantation. Unfortunately, the supply of donor organs outpaces patient demand. As many prevalent forms of heart disease, including heart failure, are associated with cell loss, the idea arose to use cell-based therapies to either repopulate the heart with new functional cells, promote proliferation of existing cardiomyocytes, and/or reverse a pathological remodeling. However, the use of cell-based therapies has been limited by 2 factors. First, studies have demonstrated significant loss of transplanted cells within the targeted myocardium within 24 hours of transplantation, where greater than 90% of the cells are lost. Second, transplantations of large doses of bone marrow cell-derived cardiomyocytes have been linked to the presence of ventricular arrhythmias. Despite these findings, experiments involving the transplantation of bone marrow cells, such as mesenchymal stromal cells, have shown benefits such as improvements in myocardial function, as well as reductions in adverse remodeling, highlighting the therapeutic potential of cell-based therapies. This brings me to the agents we will be using in our study. Our study is part of the Regenerative Medicine Innovation Project with the National Institute of Health and is in collaboration with the University of Wisconsin, Fujifilm Cellular Dynamics, and Cellular Logistics. Fujifilm Cellular Dynamics has developed a robust and scalable induced pluripotent stem cell differentiation protocol to manufacture an enriched population of committed cardiac progenitor cells, where 96.9% of the cells differentiate into cardiomyocytes under standard cardiomyocyte maintenance media conditions. Cellular Logistics provides a human cardiac fibroblast-derived extracellular matrix rich in fibronectin, which can be milled so that it can be introduced via catheter. The extracellular matrix can be combined with the committed cardiac progenitor cells and can be introduced into the myocardium. Here I am showing the characterization of the committed cardiac progenitor cells. The committed cardiac progenitor cells post-thawing are identified as being CD56 and PDGFR alpha positive. 7 days after thawing and under standard cardiomyocyte maintenance media conditions, the committed cardiac progenitor cells will begin to differentiate into cardiomyocytes as demonstrated by its expression of NKX, SM alpha actinin, and the production of cardiac troponin I and cardiac troponin T. Over on the right, I have a video of the committed cardiac progenitor cells post-differentiation contracting. The extracellular matrix is produced from culturing human cardiac fibroblasts under special conditions. These cells secrete and self-assemble an extracellular matrix as a sheet that is primarily composed of fibronectin with little collagen 1. Introduction of cell-based therapies into the myocardium can pose challenges such as requiring an invasive open chest surgery. We use the MyoStar Transendocardial system to mimic a minimally invasive treatment modality which can be used clinically. This system includes a multi-component injection catheter and various sensors allowing for real-time collection of spatial, mechanical, and electrophysiologic information to construct a 3-dimensional endocardial surface of the left ventricle. The subsequent electromechanical map can distinguish between healthy, ischemic, and infarcted myocardium based on local voltage potential mechanical motion. For our study, the hypothesis is transendocardial injections of committed cardiac progenitor cells with or without human cardiac fibroblast-derived extracellular matrix into ischemic myocardium leads to functional cardiac tissue grafts. To test this hypothesis, we designed the following experiment, which I will now provide an overview of the protocol. We began by inducing a myocardial infarction in our Yucatan mini swine using a 120-minute balloon occlusion reperfusion of the proximal left anterior descending coronary artery past the first diagonal branch. This method has been shown to reproducibly cause large infarcts in the anterior septal region of the left ventricle with a significantly reduced global ejection fraction of approximately 35%, typical of a patient with a poor prognosis for heart failure and sudden death. After the infarction, we implanted a loop recorder to monitor for arrhythmias. 4 weeks after infarction, the animals had a baseline MRI and were then randomly assigned to 4 treatment groups, being A, committed cardiac progenitor cells with human cardiac fibroblast-derived extracellular matrix, committed cardiac progenitor cells alone, human cardiac fibroblast-derived extracellular matrix alone, or control media, which was 5% flex albumin. 8 weeks after infarction, the animals had their follow-up MRI and completed the protocol with follow-up necropsy, histology, and loop recorder interrogation. As this is an interim analysis, I will be later providing the results from only 3 of the treatment arms, which I have circled in blue. All animals were loaded with amiodarone a week prior to infarction. During the infarction, the animals were treated with continuous lidocaine infusion. After the infarction, the amiodarone and lidocaine were discontinued. 2 days prior to treatment, the mini swine were then started on their immunosuppression regimen, including cyclosporin, abatacept, and methylprednisolone, in addition to being started on prophylactic Bactrim. We would also give Exceed on the day of treatment. Kidney function, electrolytes, and cyclosporin levels were monitored throughout the experiment. Upon reviewing the baseline MRIs, we were able to determine that all animals had an anteroseptal infarct. And so 8 weeks after infarction, all animals were sacrificed. The left ventricle was isolated and then divided into 3 areas. Area A covers a scar, area B covers a septum, and area C covers most of the remaining left ventricle free wall. Each area is then sectioned in the short axis, and each heart provides approximately 30 tissue blocks. This slide demonstrates how we use the MyoStar system to generate a 3-dimensional map of the endocardial surface of the left ventricle. We use the system to identify in real time the area of infarct, which is shown in red. With this information, our team is then able to use the MyoStar catheter to provide intramyocardial injections of the treatment, which is shown with a black dot. Our goal was to apply treatment to the peri-infarct region and within the infarct. At this time, not all of our animals have completed the protocol, and so final analysis is pending. There were no observed evidence of pericardial effusions on MRI, and there have been no discovered tumors or teratomas on gross pathology. Here are the loop recorder results from our animals in a couple of the cohorts. On the x-axis, we have the timeline with day 0 representing when the animals received treatments. On the y-axis, we have ventricular tachycardia in minutes per day. We saw no ventricular tachyarrhythmia events in the committed cardiac progenitor cells, the cardiac progenitor cells with human cardiac fibroblast-derived extracellular matrix, and in the human cardiac fibroblast-derived extracellular matrix groups alone. The only significant episode of ventricular tachyarrhythmia was noted in a control animal, which I have shown. Using staining for Ku80, cardiac troponin I, and DAPI, we were able to survey our tissue blocks for evidence of committed cardiac progenitor cell engraftment. Here we have an example of a graft injected into the porcine myocardium. On the left, we have trichome staining, and on the right, we have immunohistochemistry. We see within the well-defined porcine myocardium, we have a graft of Ku80 positive cells in red, surrounded by cardiac troponin I in green. This is another example of a similar graft at a higher magnification. Moving from left to right, you see the same graft bordered by the healthy porcine myocardium at progressively higher magnification. Once again, the cardiac troponin I is labeled in green. The nuclei Ku80 is labeled in red this time, and the nuclei is labeled in blue. In conclusion, committed cardiac progenitor cells can engraft after transendocardial injection into the immunosuppressed porcine myocardium. Committed cardiac progenitor cells can differentiate into cardiomyocytes a month after delivery. Committed cardiac progenitor cells have not resulted in ventricular arrhythmias a month after delivery. At this time, I would like to give thanks to the National Institutes of Health, the University of Wisconsin-Madison, Fujifilm Cellular Dynamics, and Cellular Logistics for making this project possible. I would like to give special thanks to Dr. Tim Camp and Dr. Amish Ravel for their mentorship during this project. I unfortunately cannot thank every individual member who has assisted in this project, but here they all are. And that is what I have for everyone. I will now be happy to answer any questions.